Evening viewer, welcome to our Glory Globe TV. Here's a breaking news. Yes, scientists at the University of Illinois, Chicago and Harvard University have developed an antibiotic that could give medicine a new weapon to fight drug resistance bacteria and the disease they cause. The antibiotic Cresmomycin, described in Science, effectively suppresses pathogenic bacteria that have become resistant to many commonly prescribed antimicrobial drugs. The promising novel antibiotic is the latest finding for a long-time research partnership between the group of Yuri Polikanov, Associate Professor of Biological Science at USC, and colleagues at the Harvard. The USC scientists provide critical insights into cellular mechanism and structure that help the researchers at Harvard design and synthesized. In developing the new antibiotic, the group focused on how many antibiotics interact with a common cellular target, the ribosome, and how drug resistance bacteria modify their ribosomes to defend themselves. More than half of all antibiotics inhibit growth of pathogenic bacteria by interfering with their protein biosynthesis, a complex process catalyzed by the ribosome, which is akin to a 3D printer that makes all the proteins in a cell. Ponikanov said antibiotics bind to bacterial ribosomes and disrupt this protein manufacturing process, causing bacterial invaders to die. But many bacterial species evolved simply defenses against this attack. In one defense, they interfere with antibiotic activity by adding a single methyl group of 1 carbon and 3 hydrogen atoms to their ribosomes. Scientists speculated that this defense was simply bacteria physically blocking the site where drugs bind to the ribosome. Like putting a push pin on a chair, Polikanov said, but the researchers found a more complicated story, as they described in a paper published last month in Nature Chemical Biology. By using a method called X-ray crystallography to visualize drug resistant ribosomes with nearly atomic precision, they discovered two defensive tactics. The methyl group, they found, physically blocks the binding site, but it also changes the shape of the ribosome's inner guards, further disrupting antibiotic activity. Polikanov's laboratory then used X-ray crystallography to investigate how certain drugs, including one published in Nature by the USC-Harvard collaboration in 2021, circumvent this common form of bacterial resistance. By determining the actual structure of antibiotics interacting with two types of drug resistance ribosomes, we saw what could not have been predicted by the available structural data or by computer modeling. Polikanov said, "It's also better to see it once than hear about 1,000 times, and our structures were important for designing this promising new antibiotic and understanding how it manages to escape the most common types of resistance." Mycin, the new antibiotic, is synthetic. It's organized to the methyl group interference and attach strongly to ribosomes, disrupting their function. This process involves looking the drug into a shape that is pre-optimized to bind to the ribosomes, which helps it get around bacterial defenses. It simply binds to the ribosomes and acts as if it does not care whether there was the methylene or not. Polikanov said it overcomes several of the most common types of drug resistance easily. In animal experiments conducted at Harvard, the drug protected against infections with multi-drug resistant strains of common disease drivers including Staphylococcus aureus, Escherichia coli and Zoonosa. Based on this promising result, the next step is to assess the effectiveness and safety of mycin in humans. At this early stage, the process demonstrates the critical role that structural biology plays in designing the next generation of antibiotics and other life-saving medicines, according to Polikanov. Without the structures, we would be blind in terms of how these drugs bind and act upon modified drug resistance ribosomes, Polonakov said. The structures that we determined provided fundamental insight into the molecular mechanism that allow these drugs to evade the resistance. In addition to Polonakov, UIC co-authors include Elena Alexandrova, Egor Sairogan and Maxim Seltov on the science paper, and Alexandrova, Sairogan, Sevatova and Samson on the Nature Chemical Biology paper. Then the report, Glory Globe TV, Delhi, Bharat.